Introduce a man who needs no introduction, Paul Barber from our own department. Paul is up for promotion to full professor in our department this year. So I'll give a little rundown of his history. He got a BS in Ecology and Evolutionary Biology from the University of Arizona. Did his PhD at Berkeley with Tyrone Hayes, who was here last year, where he used genetics to understand patterns of connectivity between mountain islands and tree frogs. He then went on to do a postdoc at Harvard with Steve Columbi, where he developed his independent research program in the connectivity of marine ecosystems. That was funded by NSF postdoctoral award. Paul then went on to a position at Boston University in the Department of Biology and the Marine Program, where he spent 6 years before coming to UCLA in 2008. Here, he has established, or rather continued, his leading research program in applying tools of molecular ecology to conservation and patterns of connectivity of marine ecosystems. And he's also continued to develop his very strong integration of research activities with education. And along those lines, he's had many successes in the large award and fundraising category. He got an NSF Career Award back in 2004, and followed that with a very prestigious Presidential Early Career Award in Science and Engineering, the PCASE. In 2007, got an NSF Partnerships in International Research and Education grant, and in 2009, co-founded the Indonesian Biodiversity Research Center, which promotes research and education efforts in the Coral Triangle. And that was funded by NSF and USAID, I think. So today, Paul's going to tell us about some of the great work that's been coming from that site.
Great. Thanks a lot. I always begin my talks with acknowledgments, because without the work of a lot of people, in particular in the work that we've been doing, it is a tremendous number of people who have been involved: PIs, postdocs, graduate students, a whole gaggle of undergraduate students over the years, and then a number of different funding agencies. But I always find it a bit unfair that I'm always sort of the public face of the program. And so since I can't acknowledge everyone individually, I just want to sort of let you see some of the faces of people who have worked on this and who've contributed greatly, whether they're Indonesian students, American students, graduate students, undergraduate students, Indonesian boatmen and fishermen and whatnot that have supported us over the years. I also want to acknowledge my family, because they indulge me by letting me go to Indonesia. They go with me, but we go to Indonesia for 2 to 3 months every year, and that's basically been since before they were born. So I couldn't do it without their support either. So I'm going to be talking about the Coral Triangle, and this is a region that is comprised of 6 countries: the Philippines, Malaysia, Indonesia, Papua New Guinea, East Timor, and the Solomon Islands. And it's defined by the presence of 500 or more hard coral species. And if you get into the water in the Coral Triangle, what you see is an amazing coral reef environment. Tremendous amount of diversity of invertebrates and vertebrates. And it's just absolutely teeming with life. It's just a truly spectacular place. Now, if you look at patterns of global marine biodiversity, what you'll see is that biodiversity peaks here in the center of the Coral Triangle, indicated by red. And as you move out of the Coral Triangle, there's a very sharp decrease in the amount of biodiversity. Now, this is a pattern that's been known for decades, and there are a number of hypotheses that have been put forward to explain this. The first is that this region is a center of overlap, and this draws on the ideas developed by Sir Alfred Wallace. And this is a terrestrial biodiversity hotspot, and the Asian fauna and Australian fauna overlap in Indonesia. Well, in the marine context, what they think is that there is a Pacific Ocean fauna and an Indian Ocean fauna that overlaps here in Indonesia, creating elevated biodiversity. It's also been suggested that this region is a center of accumulation. And we've got all these small little peripheral islands in the Pacific Ocean and the Indian Ocean, and it's believed that maybe speciation occurs there. And then as those species increase their ranges, they eventually accumulate into the center of the coral triangle, adding marine biodiversity to this diverse region. The other hypothesis that's been put forward is that this region is a center of origin, and that speciation is actually occurring within the coral triangle, and that biodiversity then is being exported to the peripheral islands of the Pacific and the Indian Ocean. Now, if we want to understand how the coral triangle biodiversity hotspot evolved, we essentially need to answer two questions. The first is, where is lineage divergence occurring? And the second is, what are the processes that drive that lineage divergence? This gets us into the topic of speciation. And if we think about how speciation occurs, the vast majority of it occurs in allopatry. There is a population that at some point in the past gets bisected by some sort of barrier to dispersal. And as evolution progresses over time, these populations, in the absence of gene flow, assume their own evolutionary trajectories, eventually culminating in speciation. Now, everyone in this room, as far as we know, is a terrestrial vertebrate. And as such, we view the world with a particular set of biases. And no one in this room would have any difficulty seeing the barrier to dispersal that led to the diversification between Kaibab ground squirrels and Abert ground squirrels on the opposite rims of the Grand Canyon. But if we look out onto the ocean, where are the barriers? What we see is water. What we think of is large pelagic predators, things like tuna and sharks, that can move great distances on their own throughout this massive undifferentiated environment. So where are those barriers to dispersal? The first thing we have to realize is that this is not our average marine organism. Our average marine organism are things like crustaceans, these stomatopods, or clownfish, or clams, where if they're not physically attached to the bottom, they have such a limited range that they would never, ever be able to disperse great distances on their own. So, no matter what the geniuses at Pixar might try to convince you, a clownfish is not going to jump on the East Australian current and move from one coral reef to another, okay? That doesn't mean that those currents don't come into play. Where they come into play is that dispersal, while not achieved by adults, is achieved by a larval dispersal phase. These small larvae enter into the plankton where they're acted upon by ocean currents, and that is how dispersal is achieved for most marine organisms. Now, we don't know a lot about larval dispersal in marine environments, but what we do know is that if you increase the amount of time that a larval propagule spends in the water column, there is generally an increase in the distance that propagule will travel. You travel longer, you go farther. This is generally true. It has no bearing on traveling on the 405. But in general, for marine larvae, this is true, although there are notable exceptions. There are things that disperse far shorter distances than what you would expect based on their larval duration. We know that these larvae can travel great distances. All of these black dots are capture points from plankton toes in which Rudy Scheltema back in '86 found the larvae of the adult forms of these coastal dwelling marine gastropods. So way out, all throughout the middle of the Atlantic Ocean, you're still finding the larvae of these coastal marine gastropods. Now, because these larvae travel on ocean currents, strong current should promote, far dispersal. And this is an example of the type of dispersal that you could see. This is Indonesia. This is Sulawesi here. And these squiggly bits of spaghetti on the figure here are the tracks of drifter buoys released by Roger Lucas and his colleagues back in 1991. And the time period between these blue dots is 10 days. So in 30 days, this buoy, passively drifting, traveled 1,400 kilometers. That's a tremendous dispersal potential if you were a larvae that can travel for 30 days. So that begs the question, if we have strong currents, if we have larvae that have the ability to disperse long distances, where are these barriers to dispersal? Now, the real challenge in answering this question is that unlike large taxa like sea turtles and sharks, we can't just affix a satellite pop-up tag and let the animals tell us where they're going. You know, most of these larvae are about the size of a grain of rice. A grain of rice would be very, very difficult to track in the Pacific Ocean, okay? Even if we could, even if we had that technology, out of 100,000 larvae, maybe 3 or 4 of them actually survive. So the odds of us actually choosing the right larvae to follow, basically 0. So how do we do this? We have to be clever. So let's think of a clownfish. This is a little Amphiprion clarkii. And these clownfish live in populations. So here we have 2 populations around a couple of islands. Now, these aren't just individuals. These are members of genealogies. So we have a clownfish that have offspring. Those offspring have offspring, and so on and so forth, moving forward. Now, it's not just that there's a genealogy. There are mutations that occur in that genealogy, such that we may have different lineages here. Some lineages go extinct, some make it into the present day. And so we can go out and we can look at these populations, but then look at them from a genetic standpoint, where let's say we've got 2 haplotypes, 1 and 2. Here our fish have now been reduced down to their genetic composition, and what we see is that these individual populations have equal frequencies of these haplotypes that can be summarized by these pie diagrams. And the only way that we can get this type of genetic homogeneity is if the larvae from one population are moving to the other, the larvae from one population move to the other population, and there's this larval exchange that facilitates gene flow and the genetic mixing of these populations. Now, if we look now at a situation where there's some sort of barrier to dispersal, these larvae are not going to be able to move from one population to the other. They may go out into the open ocean, but maybe they return to their home population, or maybe they simply die. But if these larvae are not making it across this dispersal barrier, eventually the processes of mutation, selection, genetic drift will operate differently in these populations, such that they have completely different genetic compositions. And so by looking at the relative similarity and difference from a genetic standpoint of these populations, we can infer the degree to which larvae are moving between these different environments. So this is where we started a number of years ago, looking at stomatopods. This is a very pretty one. This is a peacock stomatopod, Odonodactylus salaris. Any pretty photos of stomatopods in this talk are by Roy Caldwell. Any ugly ones are mine. And we started by looking at this one species called Haploscola pulchella. We sampled throughout the Indonesian archipelago, and we found these very 2 distinct genetic lineages, and populations north of the Flores and Java Seas only have this black lineage. The white lineage is only found in populations south of the Flores and Java Sea. Now, this is a very surprising result, given that these stomatopods have a larval dispersal duration of 4 to 6 weeks. They should be able to disperse at least 600 kilometers, assuming a relatively modest 25 centimeter per second. Getting feedback here. And just to remind you, these drifters traveled in 30 days, 1,400 kilometers. So we're having genetic breaks across 200-kilometer spans where the larval duration and ocean current suggests that they should at least be able to disperse 600 kilometers. So why might this be happening? Well, why it might be happening is because of Pleistocene sea level fluctuations. As glaciers built up on land during the Pleistocene, all of that water was coming from the ocean. And so sea levels dropped. And the consequence of that in the region of the Coral Triangle is that the Sunda Shelf is very shallow. The Sahul Shelf is very shallow. And so what is today a region that is mostly ocean, it was mostly land during Pleistocene glacial maxima. So we think that that's what may be going on to explain this Pacific Indian Ocean divergence that we see. But over the years, we've increased our sampling, and we started, we went out and did some sampling in Papua New Guinea and found something interesting in that there is now a third highly divergent clade. And this is a bit problematic with this idea of Pleistocene sea level fluctuations driving all of the patterns we see, because it's hard to chop something in half and end up with 3 pieces. So it wasn't just that we're seeing this in mitochondrial DNA. We actually see this in nuclear DNA as well. This is data from the myosin gene. And there is a northern clade, there is a southern clade, there's a Papua New Guinea clade, there's also some incomplete lineage sorting. But in general, these match quite well. So the important thing about Jamie's introduction when he said what I did as a PhD student is I studied frogs, which means I didn't take physical oceanography. I was not trained as a marine scientist. And so, the impacts of ocean currents and how ocean currents work was something that I didn't really consider until I had to try to think about what was driving these patterns. And there's this current that travels north and west along the northern coast of Papua New Guinea called the New Guinea Coastal Current. And this water is retroflected in a region called the Halmahera Eddy, where this water spins around and then gets jettisoned back to the east. And you can see this here in this great animation, this water just being pumped back to the east. It's not going across the Maluku Sea. Now, what's in that water? It's larvae. Those larvae have genes. If the larvae are not making it through these currents, if they cannot make it across the Maluku Sea, they cannot bring their genes, they cannot homogenize those populations. So if I knew anything about the physical oceanography of this region before I started, I would have predicted that we should have seen a genetic break here in this region of the Maluku Sea. And just to show you that, again, this isn't just something that we see in a model. Here's the rest of that figure from Roger Lucas. These buoys just spin around, and get spit out back into the Pacific Ocean. Never do they cross the Maluku Sea. So that gives us a good hypothesis to test. This clade should extend all the way over to Halmahera, where it then should be replaced by this black clade. So we went out, we sampled some more, and what we found was unexpected, and now there's a 4th distinct clade. And after I finished crying, I thought about this some more and realized that this was actually a really useful result, because what we could do for the first time was compare patterns of gene flow just within a single clade across this region of the Maluku Sea. And what we found was really cool, because there is a strong genetic break between populations in this black clade on the eastern side of the Maluku Sea and populations in the black clade on the western side of the Maluku Sea. Whereas if we look at all of these populations here, there's not a single significant FST value between any of those populations. So unlimited gene flow across 1,200 kilometers here. Strong barriers to gene flow across about 150. just across the Maluku Sea. Something's going on right here. So why then might we have this fourth clade? Well, this is probably due to the geologic evolution of eastern Indonesia here. If we go back about 3 million years, the islands that become Biak and Yapen are essentially almost closing off this entire bay, isolating populations in this region. And even now, that sill at the mouth of the bay is very shallow. So when sea levels drop during Pleistocene low sea level stands, this bay gets almost completely cut off. So that may be what is driving these patterns. But all I'm doing right now is storytelling. We have some patterns, and I'm telling you a story in terms of what I think the process might be. And how do we move beyond, just telling stories? Well, John Avis in his phylogeography book talked about the importance of shared pattern and process, and that the only way that you can get shared genetic patterns in multiple co-distributed species is through some broadly acting environmental process that's impacting everything. And so we decided we'd take a comparative phylogeography approach, and we started a program known as the Diversity Project. Two goals of this project are, one, increasing participation of underrepresented minority groups in marine science. The second is to do a major comparative phylogeography study to test the generalities of these patterns that we saw initially in stomatopods. So these are just some of the 72 taxa that we're hoping to hit. And rather than walk you through all of the results, what I'm going to do is just show you the general patterns. And in things like sea stars, clams, snails, big things like Scombroma commersoni, the Spanish mackerel gets bigger than me. We see evidence of divergence between Indian and Pacific Ocean populations. So here's the checkerboard wrasse, Halochoris hortulanus. And we have a Indian Ocean lineage, and then we have this white clade here, Pacific Ocean lineage. The Indian Ocean lineage essentially stops along the southern and western boundary of the Sunda Shelf. The Pacific clade doesn't extend into the Indian Ocean at all. We have some Indian Ocean haplotypes that do make it into the Pacific Ocean, but we've got one group on the western side of the shelf. We've got another lineage on the eastern side of the shelf. So this suggests that maybe there's something to this Pleistocene vicariance. In a number of species, from stomatopods to clownfish to, again, giant clams, we see divergence across the Maluku Sea, and this is some data by Sam Chang, who's a graduate student in my lab, who actually collected this as an undergrad in the Diversity Project. And what we see is that there's this one lineage that goes all the way up to Halmahera, and then once we cross into the Maluku Sea, it's replaced by this other lineage. The southern shore of Papua always seems to have a genetic affinity with Sulawesi, and we think that may be a function of this chain of islands here, but we haven't sampled enough in that region to be able to test that hypothesis. But this is a very common result that we see in a lot of different taxa that we look at. A number of species, from stomatopods to clownfish to clam, show both of these patterns. And we see both Indo-Pacific and divergence between the Indo-Pacific populations and across the Maluku Sea. So in the clam, we've got this one clade that's in eastern Indonesia that stops at Halmahera. We've got this other clade that extends through the central Indonesia and into the Philippines. We've got this third clade that's only found in western Sumatra. And this is based on mitochondrial DNA, but we've also looked at microsatellite DNA, and that allows us to look at these populations in a slightly different way. And just before I show you the results, because a lot of people may not be familiar with microsatellite assignment tests, essentially there's this program called Structure, And it takes your microsatellite data and it essentially says, can I split this into different groups? And you can say, define either 2 groups or 3 groups or 4 groups, and it will split your microsatellite data into these groups. So in this case, we've got 2 groups here. Then what it does is it looks at the individual genotypes of samples in your data set. And it will assign a probability of membership to one of these groups. So sample 1 has a 100% probability of being in the blue group. Sample 2 has about a 90% probability of being in the blue group, but about a 10% probability of being in the yellow. This 90% yellow, 10% blue, this 100% yellow. So what you end up with is something that looks like this, and each one of these lines represents an individual, and that individual has a probability of being, in this case, assigned to one of 3 groups, a red group, a green group, or a blue group. And when we look at where these groups are, we get some new insights into what's going on with these clams. We've got this red group, again, only in Western Sumatra, so just like the microsatellite data. We have this group as well, this blue group that's in eastern Indonesia, but it's actually in the Bay of Cendrawasih. It's not the entire it's not the entire Bird's Head region of Indonesia. Then we've got this green group, and the green group seems to be mostly Philippines. And then the central part of Indonesia seems to be this mix between this green group from the Philippines and this blue group from the Bay of Cendrawasih. So we've got these 3 groups again, but the use of this microsatellite data has refined our understanding of these patterns. Now, this pattern of isolation in Chelek Cendrawasih is something that we see in stomatopods, we see it in small pelagic fish, sea stars. There's actually about 14 or 15 new species of reef fish that we've defined genetically. There's subtle phenotypic variants in these fish. And if we compare fish inside Chelek Cendrawasih to fish outside of Chelek Cendrawasih, we see that there are really strong genetic differences between them. And so working with Mark Erdman and Jerry Allen, combining morphological and genetic approaches, there's probably been 14 or 15 new species of fish from this region defined in the last 3 or 4 years. So, just to remind you, here's this data from Haptoscope Pulchella, where we've got this distinct clade right in the middle of Chelek Cendrawasih. So, the last pattern that we see is that there's some things like particularly corals, but there's also one species of stomatopod, we've seen this, where there's a really strong pattern of differentiation on fine geographic scales. So in this case, rather than looking at the entirety of Indonesia, we're just going to look just at the Bird's Head region of eastern Indonesia, and we're going to zoom in, and each one of these little dots are collecting localities from Ceratopora hystrix. And what we see when we look at the microsatellite data in structure is that we have 9 distinct groups. And for the most part, each one of those 9 distinct genetic clusters, with the exception of 3 and 8, basically characterize an individual population. So even on very, very small spatial scales, this is 100 kilometers here, so we've got 4 populations in this region in less than 100 kilometers. And genetically, we can tell them apart, and that's because the absence of significant amount of gene flow because of the absence of a significant amount of larval exchange. So if we can just kind of summarize here these genetic results, we've got divergence in Western Indonesia. We've got divergence in the central part of Indonesia. We've got divergence in eastern Indonesia, and then even within eastern Indonesia, divergence within the Bay of Cendrawasih. Well, this suggests evidence for divergence within the coral triangle in support of the center of origins theory. So there do seem to be barriers to dispersal within this region, promoting lineage diversification. Now, that's not the whole story. That's not to say that that's only what happens. I did a study here a couple of years back with a graduate student, Joshua Drew, and what Josh did was a longitudinal study of Pomocentris molucensis all the way from Fiji to western Sumatra. And what we see is the most basal haplotypes are found in Fiji. The next most basal are in the Marquesas. The next, in Vanuatu, sorry, wrong ocean. The next most basal clade is in the central part of the Coral Triangle. The most derived clade here is in western Sumatra. So there's this pattern of dispersal and divergence from Fiji into the Coral Triangle, which is exactly what we'd expect to see under a center of accumulation model. So it's not very surprising. We've got multiple processes contributing to the formation of this pattern. We've got evidence for center of origin and center of accumulation. Now, going back to this comparative phylogeography approach by Avis, based on the evidence of these shared patterns, the shared processes that could potentially explain these that we've talked about, sea level change for divergence between Pacific and Indian Ocean, the effects of the Halmahera eddy for the divergence across the Maluku Sea. But this is a very qualitative comparison. What we'd like to do is to get beyond making these qualitative comparisons and get to something that's much more quantitative, to see, can we really truly link pattern and process? So a few years back, I started a collaboration with Jonathan Poole, who was a graduate student at the time at Erasmus in Miami. And he developed a computer model for larval dispersal throughout the Coral Triangle. And this is an individual-based model. So he had individual larvae that would be released from coral reefs throughout the Coral Triangle, and these different colors are these different release points. And these larvae would have some sort of dispersal kernel. Now, what a dispersal kernel is, this actually comes from pollen dispersal originally, but it gives you the probability of dispersing a particular distance. So here we've got dispersal distance on the x-axis. We've got the density of larval propagules on the y-axis. And in this dispersal kernel here, we've got the highest probability associated with dispersing only 15, 20 kilometers. Contrast that here with long-distance dispersal where we've got a much higher probability of things making it greater than 50 kilometers. Now, the important thing with the dispersal kernel is that there's the sort of the average. So in here, we've got a short dispersal distance, on average, but there are these long tails. So even though these probabilities may be very small, often they're not zero. And so what you can do is you can release these virtual larvae into your virtual ocean and give them various dispersal characteristics. And then you can look at how the model predicts that they move from one location to another. So here we've got source populations on the y-axis. We've got a destination population on the x-axis. And the colors indicate the probability of connecting, of larvae making it from one location to another. These dots are really small, so we'll make them bigger. Things from Sumatra end up in Sumatra with a high probability. Things from Makassar end up in Makassar with a high probability, but with a lower probability, sometimes they make it down to Flores, an island that's to the south, which makes sense based on the ocean currents. Now, this is after a single generation, but we can run this model over and over and over again, and a different pattern emerges. And what we see is that there are these blocks here in red where there's a very high probability of connectivity. And there are these regions here in gray or blue where essentially connectivity is not happening or it's extremely low probability of occurring. Now, this is a big mess. It's hard to look at, so I'm going to simplify it and ignore all the parts of that figure that are not part of the study sites that we've been talking about so far. When we do that, what we see is here's all these Western Indonesian populations. You know, high connectivity among them, basically very limited connectivity at all to the rest of Indonesia. Here's the central Indonesian populations, high connectivity among them, very limited connectivity to regions outside. Eastern Indonesia, high connectivity among them, although we have two distinct regions of high connectivity, just as what we saw in the genetic data. So this model is actually predicting these genetic patterns that we've seen in the data. That's pretty cool. That's still not very satisfying, though, because that's still very qualitative. What we really want to be able to do is say, you know, to what degree can we ascribe these genetic patterns to individual larvae traveling in the physical oceanography of this environment, given the ecological characteristics of those larvae, how long they're in the water column, what their mortality rate is, you know, do they have any sort of homing capabilities, do they have swimming capabilities, and how do we incorporate that? Well, and how do we test it to see if it matches the predictions? Well, once you move away from mitochondrial markers and you start using assignment tests, you can do some cool things. This is the results that you get from that program structure, and this is these coral data we saw earlier. But there's a different program called BayesAS Plus. It's for Bayes assessment, or a Bayesian assignment test. And what that allows you to do is look at the genetic connectivity in a probabilistic way between these different environments. So if we look at the diagonal here, what this is saying is that 98% of the samples that were sampled from Alui most likely came from larvae produced in Alui. And if we look across the diagonal here, by and large, we're looking at self-recruitment. Locally produced larvae are seeding these individual populations. That's exactly what this is saying here as well. But we can look at other populations like here, where Awi is getting 70% of its larvae from Awi, but 21% of its larvae are coming from Monmon. So what's cool about this is that we can use a different type of connectivity model. In this case, we're looking at a probabilistic connectivity model developed by Eric Tremmel, who's a collaborator on our NSF Pyre Award. And what he can do is he's developed this really cool model. All of these colors that you see are larvae that are being released into this virtual ocean. And those larvae have various different behaviors and larval durations, different ecologies. And he can look at the probability of larvae making it from one reef to another. Now, this can be done in a way where we can look at average dispersal corridors. And in this case, red indicates high levels of connectivity, green, intermediate, blue, essentially none. And we see what we saw in that other model. We've got connectivity within central Indonesia and the Philippines. There's not much connectivity between eastern Indonesia and central Indonesia. He hasn't updated the figure, but there's not much connectivity between central Indonesia and western Indonesia. But more importantly, because of the way the model is set up, he can look at individual probabilities of going from a source reef to a destination reef. And then in a graph theory framework can talk about, for all of the sites that we are working at, what's the probability of there being a successful dispersal event from population 1 here to, say, population 2. And he can, or population 1, population 2. So what we can then do is take those numbers and the connectivity matrix predicted from the biophysical model, we can compare that to our genetic connectivity matrix. We can test this using a Mantel framework and tell exactly to what degree these genetic patterns can be described by the process of physical advection of larvae in this aquatic environment given their larval behaviors. Now, the model has been finished, it's running. We have genetic data to do this from clownfish, from corals, from giant clams. We don't have the results yet from letting the model run with those individual larval parameters. And I was hoping that would be ready for today, but it's close. So we're almost there. So we're almost able to give you a number here. When Eric did this out in the Central Pacific for his PhD thesis, he was actually able to get an R-squared value of 57%, which is pretty remarkably high. So we're hoping for something similar. Now, this is going to help us link pattern and process. Another thing that we'd like to do to link pattern and process is use a landscape genetic approach to see how the ecology of these regions, the ecological parameters on these reefs, may be affecting the underlying genetic structure that we see. As its name implies, landscape genetics, it was not designed with marine ecosystems in mind, and that's because what makes landscape genetics possible is this really cool database called WorldClim, which has a whole host of environmental variables across the entire surface of the globe, terrestrial surface of the globe, so 30% of the Earth, at a 1 kilometer resolution. And that allows people like Victoria, who spoke a few weeks ago, to do really cool things like comparing genetic variation in oaks with respect to precipitation regimes in California. So, high precipitation here, low precipitation here. How does that influence the distribution of genetic variation across that space? Well, the only database we had to work with is called World Oceans Atlas, and as you can see, the Florida Peninsula and the Caribbean, you can't do anything with this. The resolution is 1 degree, it's 111 kilometers. This is totally useless to do any sort of meaningful landscape genetic approaches in a marine environment. Last year, there was a database published called BioOracle. This improved the resolution to 5 minutes, so now we're down at 6 kilometers, but no bathymetry. So if you work on tuna, or if you work on phytoplankton primary production, this might be your database. But if you're working in coral reef environments, you gotta have bathymetry. And so my student Elizabeth Jones, who's still at BU, who's finishing up hopefully in December, she decided, okay, if no one's gonna make it, I'll make it myself. And so she did, and she developed this thing called MARSPEC, Marine Spatial Ecology database. And this is equivalent to WorldClim. It's at 30 arc seconds, so that should be 1 kilometer, not 6. So you can see much more detail than what you see in BioOracle. It also has, not just the properties of the water itself, but it has these geophysical barriers that allow us to capture the bathymetry as well. And if we look at a place like Indonesia using MARSPEC, and in this case we're just looking at the water, we're looking at temperature and salinity, you can see all sorts of geographic variation and environmental conditions that could be shaping the genetic patterns that we see. And just to give you an idea of how this can be used, a couple of examples. First is, what's the role of ecology in lineage diversification? So here are 2 different species of clownfish, Amphiprion ocellaris and Amphiprion percula. If you can't tell the difference, don't feel bad. The only way you can really tell is by knowing where you collected them. All right? Why do we have these ranges ending right here? Why do they abut right here in eastern Indonesia? Well, using the actual distribution of these fish, we can train the model and teach it what are the environmental conditions under which you find these different species. And what you see when you do this is that, here's the random niche overlap distribution. Here is their actual degree of niche overlap. And so they have significantly less niche overlap than what is predicted by the model. So, okay, so we've got ecology coming into play here in terms of where we're going to find these fish. But then when you use that information and predict where you should see these fish, what you see is that, okay, Ocellaris, yep, out here in western Indonesia, Percula out here in Papua New Guinea and into Australia, but both of them should occur here in the middle. And they have different niches, they don't overlap, so that in theory they could both be in the middle. So why aren't they both in the middle? Well, it's probably because it's not just ecology, it's the physical oceanography and dispersal that is limiting the ability of Percula to move into the rest of Indonesia. So that's one way we can use this. Another way we can use this is to look at data that we have now. So here's our Haptosquilla pulchella data that we've talked about before. Again, we can train the model to predict where we should see Haptosquilla pulchella, and it actually does a very good job, except for it over-predicts up here in Micronesia and the Marshall Islands. But then you can lower sea levels, and you can ask, where would these populations likely find environmental parameters conducive to persistence? And what we see is that we have these tiny little pockets. So there's still lots of ocean here. But once you lower sea levels by over 500 feet, the other sort of geological variables, those bathymetric variables, are no longer conducive to having populations in much of the range where you see it now. And so we may have ecological differences reinforcing or in fact helping drive these patterns of genetic variation that we see. So that's kind of cool. Now, I've been talking at this point about origins of marine biodiversity, but this region of the Coral Triangle is also critically endangered from a variety of different anthropogenic stresses. And in this case, red here is not indicating high biodiversity, it's indicating the location of reef ecosystems in danger of immediate extinction. The Coral Triangle is defined quite nicely, almost, except for Papua New Guinea out here in the Solomon Islands. So the most biodiverse reef ecosystems in the world are also the most threatened. We've been very active over the years in terms of trying to work with conservation groups like Conservation International to have our results incorporated into marine conservation planning efforts in this region. In fact, Conservation International has funded us specifically to do some of this work. And some of the ways that this has been used is in terms of defining regional management areas throughout the Coral Triangle. It's been broken up into various management groups. This has been done with the aid of genetic data as well as other data on these marine ecosystems. The genetic connectivity profiles that we're creating for different species. Genetic connectivity or connectivity for marine larvae has been identified as one of the critical gaps to developing effective networks of marine protected areas. And so by looking at connectivity patterns with genetics, by having these models developed where we can predict connectivity patterns across the Coral Triangle, we can help fill this gap and help marine managers in terms of the siting and spacing of marine reserves. We can also do some really cool things in terms of looking at the effects of loss of individual populations. This is data from the corals that we looked at before. But what this shows is what happens if Mon Mon goes extinct? What happens if that reef ecosystem crashes? Well, it doesn't have that big of an impact on the amount of self-recruitment that we see in the remaining populations. Okay, so the y-axis here is how much more self-recruitment do we have. And a way of thinking of more self-recruitment is less connectivity, less exchange among the remaining populations. If we lose a population like Ambai here, we have a massive increase in self-recruitment throughout the rest of that ecosystem. What that's telling us is that this is a very important population in terms of maintaining the ecological connections that allow these coral reef populations to exchange larvae over space and time. So these are some of the ways that we've been using this data. But really, the problems of conservation in the coral triangle can be summarized in these next two slides. This is a total number of publications from 5 different common places to publish marine studies, summarizing total publications over 10 years that focus on the Great Barrier Reef, Caribbean, or Indonesia. This is the global epicenter of marine biodiversity. Not only is it just this total, but this is really troubling. This is looking at a 20-year history of number of publications per year from coral reefs and marine ecology progress series. And what we see is Great Barrier Reef, and this is just going through the roof. Everyone is studying the Great Barrier Reef. This is Indonesia. This is the global epicenter of marine biodiversity. How can we deal with this problem here if no one is working in this region? That's a huge problem. And the only way that we can address this problem is by improving local scientific capacity within the coral triangle, at the same time increasing the capacity of foreign scientists to work in this region that they've essentially ignored since Alfred Wallace left. So to do that, back in 2007, we got funded by NSF through the PIRE program, collaboration between ODU, UCLA, and Duke. And what we've done in this project is take an international collaborative approach where we bring scientists from the US over into the Philippines and Indonesia, where we work in collaboration with our local colleagues. We go into lab spaces that look like this. We show up with these mobile molecular labs. We unpack them. Voila, molecular lab. And just add people. And we do that. We have postdocs and graduate students that live there year-round and work there year-round in collaboration with our local counterparts. We have summer programs where we bring undergraduate students to work in collaboration with Indonesian students to do collaborative research on the origins of biodiversity in the Coral Triangle. This has led to the creation last year of the Indonesian Biodiversity Research Center. It's a collaboration between UCLA, the Smithsonian, and 3 partnering Indonesian universities that we've been working with now for, in some cases, like in the case of Diponegoro and Dr. Ambrianto, 7-8 years now we've been working together developing these relationships. And with funding from USAID, we've been able to develop lab infrastructure. This is based in Bali. We've got a genetics lab. We've got a microscopy lab. So no matter how people want to approach their biodiversity studies, whether it's from a genetic perspective, whether it's from a morphological perspective, they have the resources to do it. We've developed a 4-month-long curriculum of courses to teach both US students and Indonesian students and scientists how to do biodiversity research in Indonesia. We've got a scientific diving class that Mike Angara does. I do the molecular ecology and evolution class. Nancy Knowlton, Chris Meyer, Alan Collins teach, all from the Smithsonian, teach a course on biodiversity inventories. Nescent heard about what we did. They called us up and said, how can we help? And they decided that Alan Rodrigo and David Swafford would come out and teach a phylogenetic analysis course for us. And they did that this summer, and they're going to continue to send postdocs to work with us out there. And so, these courses are providing a mechanism to get researchers into Indonesia, to focus international research attention on the biodiversity of Indonesia, to build capacity within Indonesia, both physical capacity as well as intellectual capacity. And so, the goal through all of this research is to use this question of origins of marine biodiversity in the Coral Triangle, use this as a platform so that we can address this problem and increase the amount of research that's going on in Indonesia, in the Philippines, and throughout the Coral Triangle, so that we have data that allows us to help marine conservation efforts in this region so that fewer reefs look like this and more reefs look like this. Thanks.
Questions for Paul?
Yep.
Can you go back to the graph the probabilities of the larvae
Sure. This is the easiest way to do it. Which one?
The second.
Like that one?
Yeah, I couldn't see it so well, but where are the harsh blue lines amongst the areas?
Where are...
The
What's going on? That was weird. Where are... back there. So where are which lines coming from?
in the eastern Indonesia, the bottom right, it's where the harsh, the blue lines.
These here?
Yeah.
So the short answer to that is I'm not 100% certain because of the way the model is built. It's these individual blocks are incorporating a large number of different reef ecosystems, and sort of the way that he put this model together was essentially just sort of looking at connectivity. You just sort of like walk along the coastline of these different ecosystems and look at the probability of connectivity among them. And so while these patterns are pretty clear on the broad scale, when you start getting down to like individual reefs and much smaller regions within these broader geographic regions, he hasn't run the model in a way that allows me to pull that data out. That's one thing that I want to do, but it's probably going to be better to sort of take this approach that Eric Tremmel is doing, looking at using this kind of approach instead, so that we can actually get more of a one-to-one correspondence between the genetic patterns that we see and what the models are predicting. Because in this case, we can actually give Eric the precise places where we are sampling the precise reefs that we're sampling from, so that he can run the model just focused on those particular reef ecosystems, and that gives us a much better idea. So short answer, I can't tell precisely, but this method should be able to really refine that understanding for us. Yep.
So with models such as this, process in dispersal versus the process?
So one cool thing that we can do with Eric's model is the probability of dispersal is driven by essentially a two-dimensional flow field. And you essentially take a geography and you insert it into that flow field. And that flow field, that comes from decades of data that come from NOAA and NASA and whatnot. So it's real world that drives this model. But if you want to know what connectivity would be like during low sea level stands, you just throw in Pleistocene coastlines into that model. Now, there's going to be some changes in terms of the flow field because, during the Pleistocene, the amount of wind stress across the Pacific decreases because of the glaciations. So the flow field would need to be modified a little bit, but we can actually model that. One of the things that we're trying to figure out how to do, and this is sort of like sort of like all of science, you're desperately trying to finish up the step that you're on, but you're still already like 2 or 3 steps ahead in terms of what it is that you're doing. We're trying to figure out how to integrate the physical model of Eric with the environmental model of Elizabeth and have that environmental model actually shape the probability of successful recruitment. Because right now, the model that Eric has, it incorporates larval behaviors, larval duration, mortality, things of that nature. But if you could have the model, the probability be influenced by the degree to which the local environment that the larva makes it to being a suitable match for the ecological requirements of the adults, that would be some really cool feedback between these models that might actually help shape the patterns that we see. But we have to figure out how to do that. We're not there yet.
What do you think about the temporal profile of speciation? You have a glacial maximum, you have a sort of temporary all-passing for a while.
Yeah.
And so is there a way to integrate the temporal with the spatial?
Yeah, so, that's something that even in the original studies that we did on this, that we talked about, it's like, okay, probably this has a historical origin, but the fact that we still don't see much evidence of mixing suggests that there's something else going on, that we may have one set of processes initiating the divergence, we may have another set of processes reinforcing the divergence. And I think by taking the number of different approaches in terms of how we're modeling things, in terms of looking at the ecological based models, the biophysical dispersal models, being able to look at that in modern times, historical times, we might be able to start to disentangle some of that. It may be that different processes have precedence at different periods of time in terms of shaping these patterns.
Any questions? One more here.
This might be when you're predicting the environment the model extrapolating
So there's a whole field of paleoceanography where you can look at the general ocean conditions during glacial periods, during other periods in the past. All of the phytoplankton that are fixing carbon, a lot of these have calcareous skeletons, and they die, they sink to the bottom, and you can look at the isotopic composition of the skeletons that remain in sediment cores, and you can age those, and you can use the isotopic composition to essentially reconstruct what the oceanographic conditions were like. So it's a mixture of both that you can do.
Thank you all again.